this is one example file. So if you go to download, there is a folder exercise 2 and 3, session 2 and 3 exercise data. So everything we need is located in that folder for this tutorial and also for the afternoon tutorial. So the RMD file contains all the code and description, as you can see. I thought there is RStudio here. Never mind. And the PDF also is basically the PDF version of what you get in the RStudio file. So to start with, if you have R, can you please open the RMD file and set up the WD, the working directory, to whatever folder you have the files there. Okay, so if you set the working directory to where the files are, you should basically see the parameter files there. If you run this little code, it gives you the files that are in the parameter file. And what we need is the parameter files and the data file. Okay, so everyone got there? So the data that we have is a simulated data. If you open the file that says exercise x15, you will see this data set. And you will also get it in the RMD file. So this is a simulated data set for 3 generations with 1,200 in the first generation, 1,100 in the second, and 1,000 in the third generation. And so you have here the pedigree information in the beginning and the breeding values for this simulated data set. And then you have columns that come in pairs. This is the 3 scenarios, 3 different scenarios that actually we'll talk about it in the afternoon. So depending on how heterogeneous the farms are, the data set has been resimulated. So low heterogeneity, this is where farms are managed in a very good condition. So all the animals more or less experience the same challenge. Medium and high, basically this is where management is low and animals in the same farm experience a variety of different challenges. And if you run the second part of the code, which is basically after you read the simulated data, and you try to fit only a linear model, in this case for the low heterogeneity and of phenotype and the environment, you will see this figure, hopefully. So this is basically the very first picture I showed you in today's talk. So this is basically assuming that animals on average reduce performance as the challenge level increases. So this is the average line for all the animals, and each point here represents one animal. And you can see already that when farms are managed very well, more or less all the animals have the same challenge level, and each cluster is each farm. With medium heterogeneity, this is more dispersed. So we'll see in the afternoon that when we try to estimate the farm effect for this, we have less uncertainty that our value is correct. Whereas for this very dispersed, we want to kind of like accumulate everything and estimate one farm effect for this. So for now, we are going to only work with this data set. We will talk about this in the afternoon. OK, so the first example, we want to take this data set and run the random regression model with the pedigree information. Andres already told you about the parameter file. This is just another example, but I'm not going to go into details. So we want to fit this model to our data. And from the master file that you see already, we will extract this part of the data. So we want to fit average for all the animals. By default, blob F90 doesn't fit a mean. So we will have to put an extra column of 1 for all the animals. And then, because we want two environmental factors, I think this is the one that you will talk more about, Carolina, right? Fitting to, yeah. So this part, Carolina will explain to you later. But for now, we will have 6 effects to estimate. So the number of effects is 6. And our observation is at the column 4, which is the phenotype of the individuals. And so these 6 effects each correspond to one of those terms that you see in the model. The first one is the mean, which is one value at the first column. So first column, only one value. The third column is the environmental, or the x values. The random effect for each animal at column 2. So these are the ID of animals 1 to 300. And this was the complicated one that Andres talked about, how to fit a slope for each individual. So this will be nested to the effect 2. And this is yet more complicated that Carolina will talk about how to interpret this term in her model. OK. So if you run now so can everyone please try to run the model with the data set with BLOBF90 using the pedigree. So that would be random regression with observed covariates. You would need to run the section that says generating file, because we don't have the pedigree at the moment. So you need to make the pedigree file in the beginning, and also extract the data that you need. So write table the pedigree and the phenotype file. And then you get the phenotype and the pedigree. And if you cannot run BLOBF90 at all, there's a folder that is BLOBF90 output, and the solution is there. If you cannot run it at all, the solution is here, so you can just copy everything in case this doesn't work at all. And now this is the exercise that we have already done in the previous one using a genomic relationship matrix. So separating only the ones that I want for the generation 3 individuals. And the same for intercept. And now here we are just selecting the best individuals from 1 to number of selected. Let's say we want to select 10% best individuals. And this is the value we are interested in. So if you use a model without random regression and select only for best individuals, we are having a genetic gain of 0.2 for resilience, for production potential, but actually we are losing resilience. This is the example that Andres also talked about. We are only selecting for best animals. If we have a model that has random regression, and here I'm only choosing for intercept, then obviously because there is a genetic correlation, negative correlation, I'm losing resilience. But now in the next example, we can see that we can combine these two and improve both of these. I think it's enough for the morning. We can go for lunch.